Welcome to Severe Modern Academy inside the Life series. Today, we are here to test for the presence of starch, proteins, and lipids, also known as fats and oil, in the given food samples.
To perform the test for starch, we begin by collecting our bread sample. So the bread is hard, so we're going to make it a little bit moist by adding some little amount of water in our mortar. So the bread is moist. We're going to add it to our test tube. So we're going to add the water we used in making the bread moist. After it is done, we're going to add a few drops of iodine solution to our test tube. We're going to stir the mixture. You can see that something has happened inside the test tube, and there's a color change due to the presence of starch. I'm going to try it on protein to see whether iodine solution is used to test for protein. We're going to take our egg white. This is our egg white. So we're going to add it into the test tube. So as you can see, the egg white is in the test tube. So we're going to add a few drops of iodine solution into the test tube. We are going to stir this sample together. You can see that there is no color change in this test tube. So let us compare. Between our first experiment and our second experiment, we see that there is a color change in one of them. So our first experiment, we see that the color has changed to blue-black, and the second experiment has no color change. So showing that our first experiment has starch present in it, and the second one has no starch present in it because it is protein. Thanks.
I'm here to test for the presence of protein using our egg white as a food sample. To test for the presence of protein, we are going to use our xanthoprotic reagent, which is also called nitric acid. Normally, nitric acid is colorless, but when added to the food sample, which is the egg white, it forms yellow and tends to harden and form a solid. We are going to pour our nitric acid into this beaker to use for the experiment. We are going to pour 10 millimeters of our nitric acid into the beaker. Here in our beaker, we have our 10 millimeters of nitric acid. So we are going to take our test tube and add a sample of our egg white into the test tube. So next, we are going to add our nitric acid into the test tube to check for the protein. So as you can see in the test tube, after adding a nice amount of the nitric acid into our food sample, we can see that it turned yellow and also formed a solid, which shows that there is presence of protein in the egg yolk. Thanks.
I'm here to test for the presence of lipids, also known as fat and oil, using oil and ethanol. First, we'll begin by adding few drops of oil into our test tube. Now we are going to add few drops of ethanol into the test tube containing our oil. Now we are going to measure 10 milliliters of ethanol. Now we're going to add few drops of ethanol into our test tube containing the oil.
So viewers, we successfully carried out the food test using different food samples to test for starch, protein, and lipids using the iodine solution, ethanol emulsion test, and the xanthoprotic reagent, which was also known as the nitric acid. So the changes in color of these food tests observed has helped us to confirm that there's a presence of nutrients in these food samples. The practical test observed has helped us confirm these nutrients in the food samples and why balanced diet is important in our daily lives.
Thank you for watching Syria Modern Academy
Inside the Lab series. Don't forget to like, share, and subscribe for more educational videos. Bye.